So, as you all well know, transcriptomes are very complex. So they can be tens or even hundreds of transcript isoforms made from a single gene through alternative transcription start site and termination site usage, as well as alternative splicing. In addition, RNA bases can be modified by writers and eraser enzymes, and most transcripts also get a poly-A tail of variable length added to their 3' ends. And so this variation is really important biologically, so it has impact on the translational ability of those transcripts and their degradation, and also, of course, the change in the isoform can impact what protein isoform is made. So that's really interesting, sort of from a fundamental biological point of view, but defects in these processes are also involved in human disease. And then on the other hand, on a sort of more biotech, biomanufacturer point of view, you'll really want to be able to ensure that the correct tails and modified bases are present in your transcripts if you're producing something like an RNA vaccine. So today I'd like to tell you a little bit about how you can use direct RNA sequencing to address these questions. So the latest iteration of direct RNA sequencing is the RNA0004, and it's now available in store. So there's a few different ways you can prepare direct RNA libraries. You can do an RT to have a cDNA to sort of stabilize your RNA molecule. You can also do a poly-A pull-down to enrich for poly-A molecules. But the key point really is that, the bottom line, when you sequence it through the nanopore, you're sequencing the native RNA molecule. Okay, so what can you do with RNA0004? So with RNA0004, we now have a much increased output, so you can really do mammalian transcriptome scale analyses. So with a poly-A enriched library, we get upwards of 30 million reads, a little bit lower if you're using just total RNA. We also have significantly improved single read mapping accuracy against the human genome, so 96%. With RNA0004 and direct RNA in general, you also get the full transcript isoforms present from 5' to 3'. So here I'm showing a control transcript with known exons and introns, starts and ends, and you can see from the coverage and the reads that these exons and introns are really clearly delineated and separated. And we can, of course, see this also for example, human transcripts. You can also quantify gene and isoform expression with RNA0004. So here we have a plot showing a correlation of gene counts between two replicates of RNA0004. And you can see a very high correlation and also a high sort of range of expression values. So you can quantify gene expression. And also, it's important to know that direct RNA measures any RNA molecule that has a poly-A tail, so protein-coding genes, but also link RNAs that have a poly-A tail, which is shown here on the right. And now with the increased output, you can also capture those low abundance link RNAs much better than before. Okay, so you can get the isoforms, you can quantify them. What about those poly-A tails? So, like I briefly mentioned, poly-A tail lengths are regulated biologically, and that has an impact on translation. And the length might vary from 10 to hundreds of A's. And like Georgette briefly mentioned, Dorado can now estimate poly-A tail lengths from the reads, and it does this by looking at the raw signal squiggles and identifying the sort of flat signal that corresponds to the poly-A tail and estimating the length from there. So if you want to measure poly-A tail lengths from your direct RNA, simply add in your Dorado base caller call this estimate poly-A, and you'll then find the poly-A tail length in the output BAM file in the PT tag. So how well does this work? So we have a set of experimental standards with a known poly-A tail length ranging from 10 to 100 bases, and we then measure these with RNA0004. So the vertical lines indicate where we're expecting the tail length to be, and then the densities indicate the estimated poly-A tail length for each of those transcripts. And so you can see that they're in quite a good agreement. And so it's really easy to get these poly-A tail lengths from the data, and Dorado supports both the current new and the older chemistries for this. There are also some community-developed tools for poly-A tail length estimation, and they give very similar results on these tail standards. And yeah, just a quick side note to say that if you have PCR material, cDNA libraries, Dorado tail estimation also works on those. Okay, so RNA modifications then. So if you want to include RNA modifications in your base calling, we support M6A in drag context at the moment. And you can get that information by specifying the model you want and then the modified bases you want. And again, you'll find the modification information in the output BAM file with the MM and ML tags. So M6A. This is a very common modification in human transcripts, probably the most common one. And we have now a model that supports this. And so we've validated it using synthetic strands with RNA4 to see how well we can discriminate between A's and modified A's. And from this confusion matrix, you can see that that separation is very clear. This model also has very high base calling accuracy, so an error less than 4%. So you can nicely base call and map your data against the human reference. Okay, so you can visualize the modifications on IGV like we've seen in earlier presentation. So here what's shown is one human transcript on IGV with, oops, gotta go back, with the coverage and the reads, and the green points indicate the positions of those modified sites. And you can see that they co-occur very nicely with the track motifs that we've indicated. But there's motifs that don't have these modifications and others that have a lot of it, and this sort of amount really varies. So there's clearly some interesting biology going on there. We can also look at this more globally, so at a metagene or metatranscript level, which is shown here. So it's known from the literature that M6A tends to accumulate around stop codons and 3' UTRs. And you can see this well in this plot, where the red line indicates where the M6A signal is accumulating. So stop codons and 3' UTR, but we don't see this enrichment in conditions where we know there is no modification. Okay, we also have an ability to measure N1-methyl pseudouridine in contexts where all the U's are N1-methyl pseudouridine rather than your normal U. So why would you want to do this? Well, this modification is quite often used, for example, in RNA vaccines, where this modification gives sort of desirable translation, protein translation, and sort of immune response properties. So here we have another IGV track where I'm showing an in vitro transcribed sequence that's 100% modified. The coverage along the transcripts is very good, as you can see from the reads. This transcript, we also know, has a 30A poly-A tail. And with this model, we can, of course, also capture that tail length shown on the top track. So it's in the right position, and it has the correct estimated length of 30 A's. So if you were to base call this data with a normal model, you get very few reads aligning. So you definitely want to use this modification-aware model to base call the data. And also, the mapping accuracy is very low if you use the main model, obviously, because all the U's will be wrongly base called. So yeah, you'll want to use this model for this type of data. And yeah, just to highlight again that you can also get those poly-A tail lengths from this data. Okay, so where are we going with this? So we want to enable an easy analysis of these RNA modifications and poly-A tail lengths in the context of those transcript isoforms and also biotypes, so different protein coding and non-coding genes. We're also working to sort of diversify or extend the features we can measure. So broader or different types of RNA modifications, also RNA modifications and different types of poly-A tails as well. And then finally, we'll want to sort of enable an easy analysis of the modified bases and tails within, so have this integrated within the epitome system, so within the existing transcriptome workflows in the epitome system, so that it will be very easy for all users to start exploring the biology of these modifications and tail lengths. And with that, I'd like to thank all the R&D teams involved in developing direct RNA sequencing methods, and I would like to thank you for your attention.